Hey guys, welcome back to a new video by Baji with Xuan. So today we are back with another Cambridge AS Biology 9700 for chapter 1 cell structure on 1.2 cells as the basic units of living organisms. So this chapter is a little bit similar if you have been studying for my GCSE, and it will dive a little bit more in depth on like the basis of like the different organelles inside and the parts inside of the organelle itself that actually comprises its function. So in this chapter, we will really dive through from pretty much on every single organelle that we have learned in both animal and plant cells, and at the same time we need to know the basis of like in a microscopic level, okay, in some of the things here. So if you have any questions, please feel free to comment down in the comment section below, and yeah, hope you enjoy this video. Okay, so today for the table of contents today, there will be 5 things I'll be covering, which is eukaryotic structure and functions, animal and plant cells, vital role of ATP, prokaryotic versus eukaryotic, and lastly, viruses. So firstly, we look at eukaryotic structure and function. We need to make sure that you all recognize the organelles and other cell structures from the eukaryotic cells and outline their structures and function. Okay, so some recap I would say is that cells are divided into 2 types, which is both eukaryotic and prokaryotic cell. Eukaryotic cells are generally almost pretty much commonly seen in everywhere. Prokaryotic is mostly with mainly what we remember is bacteria as one example. Okay, but I'm sure you have in your head you have know the difference between how a eukaryotic cell looks like versus how a prokaryotic cell will look. So the difference is that the eukaryotic cell itself, which is the diagram that I'm showing you here, is that it's so much more complex. So you can say that its ultrastructure of the eukaryotic cell is very complex comparing to the prokaryotic structure. Look at the way how it's positioned, how there are so many different types of things that's comprising this cell alone, shows that how complex it is as a cell on its own. So the cytoplasm of the eukaryotic cell can be divided into membrane-bound compartments called as organelles, and eukaryotic cell can be more compartmentalized. Okay, so we'll look through a bit more on this later on. Okay, now let's look at the overview of 15 organelles. So in exam, I think something to take note here is that do not use short terms such as RER for rough endoplasmic reticulum or SER for smooth endoplasmic reticulum in the exams, and make sure you write out full word by word. Okay, so like how we've gone through this, so now we'll look at the cell surface molecule. So this cell surface membrane, right, what they do is that they actually separate the internal and the external environment of the cell. That's why the main function of the cell membrane is that it helps with the movement in and out of the cell. So they help to control exchange of cellular materials. So it's partially permeable and it forms from a phospholipid bilayer. So what is a phospholipid bilayer? It means that phospholipid is the main constituent here with both a phosphate group and a lipid group, and the bilayer means it's a double layer. You can see one of it is facing up, another facing down, another facing up, facing down. So that's a phospholipid bilayer actually. And then they move with the exchange of materials. So now, looking at the nucleus, the nuclear envelope, and the nucleolus. So they are found in all eukaryotic cell. And you can see the nucleus and cytoplasm right itself. They actually have a double barrier separating them. And I'm sure you know the nucleus inside here, they contain a lot of DNA. And you know DNA is from chromosomes is coming from DNA and protein, then they form as a chromatin. And the nuclear membrane itself, right, later on, I think, not sure if there's some diagrams here, but it's like they have a lot of pores. So you can see these pores are located here. Okay, these are all small little holes, and what do they actually do is that they will actually create a passage for them to send materials out from the nucleus itself. And inside the nucleus, there will be a nucleolus. Okay, another mini nucleus inside. So most like the more of the concentrated part of the nucleus in there. Okay, now moving on to the rough endoplasmic reticulum. So the endoplasmic reticulum is made of a series of membrane that form flattened sacs within the cells of the cytoplasm. So this is the nucleus, right? So these are all flattened sacs that are basically what is surrounding the nucleus on its own. So it's linked with a nuclear envelope like what I mentioned, and there are two ER types, with rough endoplasmic reticulum and smooth endoplasmic reticulum. So looking at the rough endoplasmic reticulum is that you can see there's so many layers of these membranes are actually on top of each other actually, and then they actually surround the nucleus. So what do they actually do is that this rough endoplasmic reticulum, they have a lot of these studded ribosomes. You can see those small little dots here are basically studded ribosomes. And these ribosomes, what they do is that they will help to process the proteins, which actually after the ribosome has done its job, this rough endoplasmic reticulum will help to basically process them properly. Now, moving to the smooth endoplasmic reticulum, the difference you can see is that you can see here in the rough endoplasmic reticulum, there's so many ribosomes, right? In smooth, there is none. But what is their role? The role of these two reticulums are two different things. For the role of the SER, they are mainly playing the role of producing lipids, steroid hormones like estrogen and testosterone. So these are some of the things. They are more fat mediated, but you can see in the rough endoplasmic reticulum is more towards the protein, because that's where all of the processing, the packaging will happen at the rough endoplasmic reticulum, and the smooth is just what is actually outside, and then they just focus mainly on the production of the lipids as mentioned. Now, we come to this part known as the Golgi body. Golgi body can also be known as a Golgi apparatus and a Golgi complex. And these are all interchangeable terms, so no worries if you can write these in exam. So they look a bit like a rough endoplasmic reticulum, you can say that also. And they actually contain a series of flattened sacs of membrane, and they can be clearly distinguished from the ER as it's not a connected to other membrane-bound compartments. So this is actually one of the big clue here, that what makes Golgi body different from the two reticulums that we learned is that they are not connected with any membrane-bound compartments like the nucleus and anything like that. So as you can see here, it's located so isolated away from this part. This is smooth, this is the rough, and this is the nucleus, right? They are completely separated from them. So these are just like an entire isolated part of it. So what they do is that they actually modify proteins and package them into vesicles. So you can see vesicles are like a small little bag, and then they contain something inside. These are like small little like coverings, okay, I would say, and they actually form this Golgi body. Now, coming to the mitochondria, which I think is something that we are all familiar with, is that they are large organelle surrounded by a double membrane. So double membrane is like you have a two-layer membrane, the inner and the outer membrane for this case. So this inner membrane will be folded. You can see the inner membrane here is folded, it's been folded here, and then they actually form what we call as a cristae. And this cristae, what they do is, again, you can see when they are folded, it usually will relate something about increasing the surface area, correct? So for here, they will actually help to do aerobic respiration as mentioned, because you know that this actually releases ATP. These mitochondrial matrix, they actually contain enzymes needed for aerobic respiration. You will know in the glycolysis and then the Krebs cycle, they involve a lot of enzymes, and these are all found in the mitochondria to be more specific. And there in this mitochondria, they actually have their own DNA, I would say. They're known as a mitochondrial DNA, and ribosomes are also found in the matrix. So what they do is that they actually help to produce proteins that are required for respiration. That's pretty much it actually. Okay, ribosomes. So ribosomes, like I said, is either isolated on its own or is studded or attached towards the rough endoplasmic reticulum, and is found from the nucleolus. So each ribosome is complex of a ribosomal RNA and also protein called rRNA. And in prokaryotes and eukaryotes, they both have ribosomes, but they are in different types of ribosomes, I would say. And eukaryotic cell, always remember that eukaryotic cell is always small, so therefore the number is high, it's 80S ribosome. While prokaryotes, mitochondria, chloroplasts, they have 70S ribosome. And what do they actually do? They do more for the translation process. Translation means that they actually, in the form of mRNA, it now translates into a protein. They're going to make a protein out. So that is actually what we know as translation during protein synthesis. In IGCSE, we don't really dive in depth on translation that much, but over the next few chapters, we'll actually explore more on the protein synthesis in detail. Now, coming to vesicles, like how I say vesicles, we can say they are formed from the Golgi bodies and the Golgi apparatus. So they are small membrane-bound sacs they used for transport and storage. So one thing you can see, something that you can relate to this is that, remember the neurotransmitters in the vesicles itself? And then what they do is that in order to allow the neurotransmitter to diffuse across, it has to fuse to the membrane. So for this case, it's the same thing. When they fuse to the membrane, then they undergo exocytosis. Exocytosis means they allow the material to exit like this, exocytosis, because exo means exit, endo means enter, so they can fuse into cell membrane to undergo endocytosis, or sometimes it can bud from a membrane during endocytosis. So it's actually working both sides, but mainly what we know from vesicles that they actually do more of the exocytosis function actually, because they will actually fuse to the membrane and then they will release it later on. Okay, lysosomes. Lysosomes are like more towards enzyme that actually undergoes more of the digestive function. They need to digest waste material, something like that. So they actually, what they do is they break down biological molecules like waste material, worn out organelles, engulf pathogens. They're basically like the janitor or the vacuum cleaner of the cell. Okay, I would say that would be a really nice way to describe it. So you can see they have a really nice membrane, a digestive enzyme inside, so they will be the one to be digesting these materials because they have enzymes. They are similar to our pepsin, all of these enzymes that are found in our body. It's the same way as how this will work. Okay, coming to more of the structural organelle is that they help to withstand the backbone of the cell itself is the centrioles. Centrioles are made of hollow fibers. Hollow means that they just looks like they have this sort of very distinct shape, and then they're made of microtubules. Microtubules are like tiny tubes, okay? They're actually going to act like a tube. That quite microtubes, right? So two centrioles at right angles that each other form a centrosome. So what they do is that these centrioles actually form on this part of it, and then they form a centrosome. And which organizes the spindle during cell division, and they're not found in flowering plants and fungi. So what they do mainly is during cell division, like mitosis, meiosis. They have something like this on the end of the cell. Now, microtubules, so like what I mentioned, they're like hollow tubules made of alpha and beta tubulin protein. So to be more specific, they're really, really small, and then they combine to form dimers, which are then joined into protofilaments. So from alpha and beta tubulin, they will form a filament. Heterodimers, they are dimers that which are different basically, and they form one protofilament. And this many protofilament will join to form this cytoskeleton of the cell, which is basically the microtubule that we are familiar to look at. So which is this tube. You can see this entire thing that's formed from many alpha beta tubulin protein. And what they do is that actually, because they act as a structure, so they do a lot of the supporting and the movement of the function of the cell inside. Okay, cilia, more of a specific type of cell which actually have hair-like projections. You can see this in ciliated epithelium in the lungs, or could be in the intestine, and they are made up also from microtubules. They are found at the surface of some of the cell, and then what they do is they allow the movement of substances over the cells, so they help to beat and may help to move the things away. That's why you can see this mainly in the lungs, the ciliated the epithelial airway inside the airways of our lungs that help to sweep dust and materials like that. So that is basically the cilia. Microvilli will be more of a specific thing that you can see in the intestine. Okay, so they are basically cell membrane projection that increases the surface area of absorption, and they found in all parts of the body that carry out absorption. So like in the small intestine, kidney tubules, and be careful not to confuse microvilli with villi, because villi is the entire thing, when microvilli is each of the small one that is found on the enterocytes, which are basically the cells of the intestine. Okay, now coming to plants, which are cell wall, outside cell surface membrane, so over structural support to some type of the cell. So this cell wall, they are made out of cellulose. Mainly, these are their main material, cellulose. But in fungi, they are made of chitin. Yeah, chitin, chitin. So these cell walls are freely permeable and do not play a role in any movement of substance, because movement of substance in and out of the cell is by the membrane, not by the cell wall. The cell wall just actually moves like a structural support again for the plant to withstand certain pressure, like let's say during osmosis like that. So that is how they actually do in the cell wall. Now, chloroplast is a little bit like a mitochondria. Okay, they also have a surrounded by the double membrane. They have their own DNA. They have their own ribosomes. So they are also made of membrane-bound components called a thylakoid stacked together to form a structure called grana. And these grana are joined by lamellae. So these chlorophyll are found in the thylakoid membrane. I will show you a bit more about this because it looks a bit confusing. So what you can see here is that this is the chloroplast. You have the outer membrane, you have the inner membrane, which actually reminds you of the mitochondria, because it's a little bit similar actually to the mitochondria structure itself. It has a double membrane. So what you can see here, the stroma is basically the liquid or the fluid that's surrounding them. So one of these, what we call as a stack, is called as a thylakoid. And when there's actually a stack of it, you call it as a granum. So thylakoid is representing one unit of this, one unit, and granum is representing the entire stack of the thylakoids. And the lamella is the one that connects between one granum to another granum like that, and then you can see it's all connected with each other. And the lumen is basically what's inside that is actually containing a lot of the chlorophyll that we know that's found inside the thylakoid. Okay, plasmodesmata is more like a bridge. It's more like a connection, I would say. And it's just basically a way to connect neighboring plant cells which are continuously, and they allow the transfer of substances between plant cells from one side to another. So it's known as a plasmodesmata. Okay, in the plant cell vacuole, you will see this known as a large permanent vacuole, which are found in the plant cell. They store what we know as a sap, okay, cell sap, and then they actually provide also structural support. And then for the vacuoles, they are sometimes found in the animal cell, but this will be very small and temporary. But vacuoles are surrounded by what we call as a tonoplast. A tonoplast is like a membrane of the plant cell. Okay, so vacuole, a membrane of the vacuole. So they call it as a tonoplast, which again is like a partially permeable membrane, like our cell membrane basically. Okay, now, if there's I kind of run through this, but to be fair, for me to really ace this chapter is to really know what are the key things, what are the key characteristics for each of the things here. You can see every part of the cell, like mitochondria and like chloroplasts, they have very significant and similar functions. It's not similar functions, similar structures in terms of like their inner membrane. They got a double membrane. They have their own ribosomes. Okay, they have their own DNAs also. So that's actually the significant difference that you should find out from this subtopic on its own. Now, coming to animal and plant cell, I hope that you can able to describe and interpret photomicrograph, electron micrograph, and also compare the structure between them. So you can see that electron micrograph for an animal cell is very complex. You can see just from this micrograph itself, you can see how complex this is. So what's the reason that we want to do this? That we want to identify organelles in the micrograph to see the shape, the location, the size, and also what is their relationship with other organelles. And same thing for the plant cells also is to see the same technique as animal cell. And for plant cells, right, they are large and similarly empty spaces inside cells will be vacuole. So you can see the big space actually covering at the center here. These are the vacuoles basically. And the nucleus is much of the darker region, and chloroplast are the next largest organelle, and then the grana is often visible. So you must know that chloroplast is the second largest organelle in the plant cell. Okay, so you can see the category, or you can see the comparison here. You can see all of these things are all things that I've categorized here, which are based on the structure common to them, and also the plant cell specific structures and the animal cell specific structure. I think you can have a look of it here and know the difference between them, because plant cell, we have their own specific thing, like cell wall, vacuoles, chloroplasts. These are all from IGCSE or from your previous education, that these are all plant cells that you can only find from that. You cannot find from animal cells. And these are the things that can compare or classify them in a very much nice way. Okay, diagram from Save My Exam here shows a really good one on like giving you the whole big picture of the eukaryotic cell. And please do have a look, because I think it's a really nice diagram, and I won't really dive into deeper again on this. And the same thing for the plant cell here. It's actually really nice to see this complex diagram here to really compare on the difference of the structure on its own. Okay, now we'll go to ATP. So the vital role of ATP, you know that ATP is produced from respiration, right? Where coming from respiration is from the mitochondria. So now ATP is basically known as adenosine triphosphate. So it's basically an energy currency for a living organism, because all living organisms need ATP in order to survive basically. And then for it, mostly what they do is actually for catabolic and also anabolic process, which catabolic means that you will be breaking down something, and anabolic is where they're going to make something. And ATP is one of the very key things that you can see as a product of respiration. And ATP is also a nucleotide, because some of the structures which you can see here is actually very similar to what you can see in a DNA. So yeah, you look at it as quite interesting, and you should try to have a look on the difference between it. Now, we'll come to prokaryotic versus eukaryotic to know the difference of the key structural features of it here. So for the prokaryotes, right, you can see they're unicellular, 1 to 5 micrometer diameter, contains a peptidoglycan cell wall. I'm sure you know about this already. Circular DNA, cell DS ribosome, like I mentioned just now. For eukaryotes, it will be ATS, and they have absence of organelles with double membrane, so they have no nucleus, no mitochondria. So for here, they have no double membrane structure, but you can see in eukaryote, they have a double membrane structure, which is basically like the mitochondria for one very good example here. Okay, this is the comparison, or like the comparison table between the two of them. You can see how big of a difference it is. I think I'll just highlight some of it. So you can see genetic material here is a circular chromosome, but eukaryotes we have linear chromosome, which are found in the nucleus. And for cell division, you know that by prokaryotes, by this binary fission, because they have no spindle fibers, they have no these centrioles or these microtubules that we had. But for eukaryotes, they have mitosis and meiosis, and you know they are separated by the spindle fibers. Prokaryotes, they have 70S, while eukaryotes have 80S, and you can see the difference between the organelles. Most of our organelles in the eukaryotes are multiple membrane-bound organelles, but for prokaryotes, they have none. And the cell wall, you know that it's made of peptidoglycan for prokaryotes, and for plants, they're made of cellulose but chitin in fungi. So I think that's just a quick summary between the difference between the two, and you must be able to list the difference, okay? Because if you do not list the difference, you won't be able to see the key difference between these two different kind of cells, because they look very different structurally also. They have some things that can be similar within themselves, but you can see the eukaryotes are much more complex and have better complexity comparing to the prokaryotes. Lastly, viruses. Viruses, you must know that they are non-living structures. They basically take advantage of the host. Let's say like viruses that affect us, they live inside us really, really well because they just need a host in order to survive. So viruses, they actually have no cellular structures, and they are more towards like a particle because they don't achieve the criteria of Mrs. Grant. Okay, they don't achieve Mrs. Grant properly. That means the movement, respiration, blah 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 like that. They contain an nucleic acid core, okay, which either could be a DNA or could be an RNA core, okay, and these are basically like their genetic material inside them. They have a protein coat known as the capsid, and some viruses, they may have an envelope. An envelope is like almost like an extra layer to protect themselves, which is also made of membrane phospholipids, and can only reproduce by infecting living cells. So they have to meet the host, and then when they're able to basically integrate what they have from their DNA into, or let's say their nucleic acid core into the host, then they're able to survive because they're able to infect very easily. And they can use attachment proteins like almost like that very good adhesion type of things, and they can very easily bind to the surface and also infect very easily. So this is how it looks like in a schematic diagram. Well, I think if you remember from IUCSE biology, we did touch on this a little bit, not so much, but now we go a little bit in depth, but not so much also actually. It's just that you just be very much familiar with the previous subtopic. I think that would be great. And yeah, if there's nothing, any questions for me? If you have none, then thank you so much. So again, reference to save my exams for all the really, really nice pictures. Please have a look on how this looks. And then it's not really a difficult chapter because some of the basics have been built already. It's just that in the second subtopic, if you learn some new organelles, then that may be something that you have to know, okay? So yeah, if there are any questions, feel free to comment down in the comment section below, and I'll see you in the next subtopic. Bye bye.